to the organizers for giving us the opportunity to present. And thanks to everyone for attending. So today we'll present some recent results highlighting the kinds of heterogeneity that manifest within bipolar disorder. And our hope is that through discussion and future collaboration, we'll not only better understand the emerging structure of this disease, but also develop better strategies for risk prediction, treatments, and therapies. More broadly, we expect these developments to highlight the importance of considering heterogeneity in the first place, especially when it comes to complex psychiatric disorders. And as a brief outline of the agenda, I've listed the speakers below, along with a few buzzwords cribbed from the abstracts. First, I'll kick things off by illustrating a few of the ways that heterogeneity might manifest at the genetic level, using data from the PGC-BD as a case study for this genome-driven analysis. Then, assuming we'll see exactly, assuming no connection issues, then Tracey from UCL and NIMH will guide us through an analysis from the opposite direction, starting with subphenotypes and delving into what makes them distinct genetically. Katie from Dalhousie will discuss some of the important relationships between phenotypic heterogeneity, symptom structure, and treatment response. And finally, Abe, also from Dalhousie, will take us home by discussing a framework that can help us categorize and understand heterogeneity more generally. We'll each aim for about 10 minutes, giving us 10 minutes at the end for discussion. And just as a guideline, please type questions into the chat. If the questions can be answered easily, the speakers will respond as soon as they get a chance. And for more involved questions, we'll address them at the end during the general discussion I mentioned, when all the speakers can participate. Now, for my part, I wanted to present a quick summary of the tools we've been working on for heterogeneity analysis, highlighting our recent results and pointing towards some of the directions that we hope to take this work going forwards. As an overview, a basic application of these strategies is shown here. So imagine you have a data array with some, can you guys see my mouse? Probably, right? Yeah, all right. Imagine you have a data array with some number of subjects, each associated with a variety of measurements, for example, genotyped SNP values. This particular array doesn't appear to have much structure within it, and a standard principal component analysis won't reveal much. Projecting the subjects onto the principal components of the data array produces a distribution that just looks like a blob. However, this data array is actually heterogeneous, and within it lurks what we call a bicluster. That is, a subset of subjects that exhibits a specific, although slightly noisy, genetic signature that involves only a subset of the SNPs. This bicluster isn't apparent initially, but by rearranging the subject rows and the SNP columns, you can see it up here. Now, given that you've found the bicluster, you can project the patients onto the principal components of the bicluster, at which point you see that there actually is some structure in the data, and it stands out if you look at the data correctly. Now, most of our recent work builds on top of our computational strategy for finding these kinds of biclusters. And while I don't want to dwell on the technical details, I can summarize by saying that we've developed a method called loop counting, which can find hidden biclusters within large data arrays. The basic algorithm iteratively scores the subjects in the SNPs by accumulating information associated with submatrices called loops, and then removing those subjects and SNPs with low scores and repeating. This process is essentially guaranteed to find sufficiently large biclusters exhibiting correlated patterns and doesn't require a parameter search or fine-tuning. So while I'll gloss over the math here, there are two features I want to emphasize. First, because the calculations involved are relatively efficient, we can use permutation tests, which I'll show later on, to assign p-values to the existence of heterogeneity and to identify which biclusters are statistically significant. Second, and perhaps more importantly, the loop counting method offers a natural strategy for contrasting one group against another, for example, cases against controls, and for correcting for covariates of various types. Now, if anyone is interested, I can link you to the details in our paper, but for now, I just want to mention that this loop counting strategy can be applied in a variety of scenarios, both to GWAS data and to gene expression data. Now, previously, I mentioned an important property of our algorithm, which is control and covariate correction. And this allows us to search within a data set containing case and control subjects and search for biclusters that exist within the case population that don't span a similar fraction of the controls. That is to say, we can define cases to be subjects with BD and controls to be subjects without, and search directly for case-specific heterogeneity. And this idea is the basis for one of our recently published papers with the PGC-BD. And now, as a quick bit of background, recall that BD has a strong heritable component, but it's been difficult to pin this down. In the hopes of illuminating the genetic drivers of heterogeneity, subphenotype-specific analyses have been run based, for example, on inter-episode remission or psychotic symptoms during mood episodes or comorbid anxiety disorders, etc. And as Tracey will tell us later, one strategy is to start with these clinically determined subphenotypes and try and trace them back to some driving subphenotype-specific genetic signatures, similar in some ways to supervised clustering. One challenge to this approach is that it's hard to get access to subphenotype data. That is, it isn't always easy to collect the clinical subphenotypes for a large set of patients in a uniform fashion, and it can be hard to obtain this data post hoc. So in this paper, we adopted a genetically driven approach, and our idea was to sidestep the complexities associated with defining and harmonizing clinical subphenotype data, and instead try and identify genetically driven subtypes directly within a training cohort. In this context, a genetically driven subtype refers to a subset of BD subjects which collectively exhibits a genetic pattern that's not expressed as strongly across a similar fraction of the controls. You could say a BD-specific bicluster. So as a pipeline, we started with only genotype data and used our heterogeneity analysis to search for a BD-specific bicluster. As a proof of principle, we used a single large study called BDRN as our training cohort. As you can guess, we did indeed find a BD-specific bicluster within the BDRN cohort, and to provide a glimpse under the hood, we assessed the statistical significance of this bicluster, or the analysis in general, by using permutation tests. And here's one example of such a permutation test. So shown in red here is the sequence of total loop scores obtained when applying our loop counting algorithm to the cases and controls in BDRN. And the horizontal axis corresponds to the iterations of the loop counting algorithm, starting with all the subjects and SNPs over on the left, and progressively eliminating subjects and SNPs as we move to the right. The vertical height of this red curve shows the average level of covariate-corrected differential expression between the remaining case subjects at each iteration and the controls. The subplot on the left illustrates the progression of the algorithm by iteration, while the subplot on the right shows the same picture rearranged so that the horizontal axis refers to the number of case subjects remaining. Shown in black are the analogous sequences obtained as trials of a permutation test where we randomly shuffle the case and control labels while respecting the ancestry-related covariates that we corrected for. And you can see that in the case of BDRN, the original run in red stands out from the cloud of trials drawn from the label shuffled null hypotheses in black. And this means that this original run is identifying case-specific heterogeneity, which is significant within the training cohort. And we can use the local z-scores to pick out the most significant iteration, for example, this one, to define where the bicluster starts. Now, as I alluded to above, this bicluster, this subset of BD subjects picked out here, was strong and clear enough, I guess you could also say here, was strong and clear enough that it replicated across the other 3 cohorts we used for validation, spanning an additional 16,000 cases and 25,000 controls. And now, given that our analysis had identified a replicable BD-specific bicluster, we could take another step. For example, by considering the individuals in the bicluster, that is, those delineating the subtype within the training cohort, and thinking of them as subtype-specific cases, we could use those subtype-specific cases to define a subtype-specific polygenic risk score, or PRS. Well, what was particularly promising to us was that this subtype-specific PRS, defined only using the bicluster cases within BDRN, was more powerful than the population-wide PRS defined using all the case subjects in BDRN, with a higher associated liability score that required fewer SNPs to reach its maximum. So let me pause and dwell on this for a moment before I wrap up. A typical assumption underlying GWAS is that given a data set, it's best to use all the cases you have access to. And in a vacuum, that should give your analysis the most power. But what we're seeing here, actually, is that there's enough strong heterogeneity in the data that by focusing only on those specifically chosen case patients that share a disease-specific genetic pattern and setting aside for the moment the other outlier case subjects that weren't part of the bicluster, you can actually improve the performance of the PRS, both in terms of the overall risk prediction and in terms of which SNPs are identified or attributed larger effect sizes. Moreover, this is my last slide, we can also apply similar logic in the opposite direction. So we can use the other cohorts available from the PGC-BD to define a high-powered PRS while leaving out the BDRN cohort. We can then test this high-powered PRS on BDRN, yielding a covariate-corrected AUC of about 72%, shown here. The immediate question, of course, is whether or not this high-powered PRS interacts with the BDRN bicluster that we delineated earlier. And shown here, you can see the covariate-corrected AUC recalculated. So this graph is supposed to mirror this one down here, which is actually just a copy of this run that I showed you before. Okay. And so you can see the covariate-corrected AUC recalculated as we restrict the case subjects to those identified by the loop counting algorithm on the previous slide. And note that as the bicluster emerges, the covariate-corrected AUC increases quite dramatically. And put in other words, when we restrict to the BD subjects in the bicluster, the high-powered PRS does even better, significantly increasing the covariate-corrected AUC and increasing the liability score by about 40 to 50%. So we feel that this demonstrates that not only does the bicluster that we found in BDRN interact with the high-powered PRS score, but it looks like it could even be one of the drivers for risk prediction. So motivated by these successes, we expect the pipeline mentioned above to yield even better results when applied to larger data sets. And so we want to apply similar analysis to more recent, larger cohorts. One important issue that we brushed under the rug is that while we've been discussing BDRN, since it was the single largest study we had access to, the other studies we looked at also had strong replicable biclusters, both in their case subjects as well as in their control populations. Some of them are shown over here, actually, involving pooling multiple studies on the same platform and searching for, in this case, control-specific biclusters. You can see that the significance values are pretty extreme. So we believe that by better delineating these heterogeneous genetic signatures and determining how they interact, we can build a better picture of the genetics underpinning bipolar. And we also want to see if the genetic structures that we've already found interact with any of the subphenotypic data that's already been collected. And hopefully Tracey can introduce us to that. Now, let me stop my share here. Oh, and there's a question in the chat. Maybe we'll wait until the end to address it. Okay, thank you. So if Tracey's there, do you want to try and, oh, excellent. All right, let you take over.
Thanks, Adi. We're happy to share our recent work demonstrating the genetic underpinnings of clinical heterogeneity in bipolar disorder, BD. Although BD represents a single label, individuals display a wide range of clinical presentations, illness trajectories, and treatment responses. Recognizing and understanding this variability is essential. Our research identified distinct genetic signatures of homogeneous subgroups within BD, referred to as subphenotypes. Knowledge of these unique genetic architectures may provide us with additional therapeutic targets, better diagnostic strategies, and more personalized interventions. Our study integrated clinical data and genetically driven clusters to model the complex relationships between clinical presentations and genetic factors. Our research began with examining dimensions of clinical variability. We observed psychotic features and comorbid conditions were key dimensions. The hierarchical subphenotype plot shows how 11 subphenotypes cluster together. Confirmatory factor analysis indicated that a 4-factor model best fit the clinical data. We used clinically anchored genome-wide association studies, GWAS, to identify subphenotype-specific genetic variants, genes, and biological pathways. Our work also incorporated findings from related disorders such as schizophrenia to explore distinct transdiagnostic factors. Our analysis demonstrated different clinical presentations had distinct, albeit overlapping, genetic foundations. We collected subphenotype data from around 25,000 BD patients. These included those with BD1, BD2, schizoaffective disorder, with varying features such as psychosis, unipolar mania, rapid cycling, panic, OCD, substance use disorder, and a history of suicide attempts. This network plot is an example of the global genetic correlations we examined between the BD subphenotypes and some 1,400 other psychiatric, cognitive, and disease traits. These correlations and heritabilities listed in the table were estimated using subphenotype-specific GWAS. Subphenotype-specific genetic variants represented heritabilities ranging from 2 to 26%, indicating there may be substantial variation in genetic influence. Although insightful, this method represents a single dimension without access to the genomic regions driving these complex correlations, which our subsequent methods help to address. These individual plots represent the results of our local genetic correlation analysis of 10 subphenotypes, offering a direct view to mixed effects in genomic regions masked at the global level. Each plot shows the local genetic correlations between a specific subphenotype and related traits. The analysis indicated mostly concordant effects in red with previously related traits, while discordant regions are indicated in blue. Labels in the plots show the traits with the strongest effects in each subphenotype. This analysis helped identify specific genomic regions with shared and non-shared genetic factors across subphenotypes, offering additional insight into these complex traits. We next performed principal component analysis, PCA, on significant variants identified in subphenotype-specific GWAS to evaluate genetic relationships amongst the subphenotypes. Closeness of the subphenotypes in the PCA plot indicates higher sharing of genetic variants related to psychosis and comorbidity. Subphenotypes near the vertical line are linked to psychosis, while those near the horizontal line relate to comorbidity. We identified 4 main clusters: Cluster 1, BD1, Cluster 2, schizoaffective disorder and psychosis, Cluster 3, intermediate comorbidities, substance use and suicide, and a distant 4th cluster comprising BD2, rapid cycling, OCD, panic, and unipolar mania. Mania likely differentiated BD1 from the schizoaffective psychosis cluster, unipolar mania, traditionally associated with BD1, located to the 4th cluster. This suggests potential stronger genetic links to instability in affective states and anxiety-related features. The heat map and ANOVA results show that local genetic correlations, less affected by sample size, significantly aligned more within PC clusters than between them, suggesting a biological relationship rather than one due to chance or sample size. These higher within cluster values provide support for the PCA analysis after mitigating potential GWAS power biases. We considered alternative sensitivity analysis such as downsampling variants and using only their intersections. However, these could introduce bias from the loss of subphenotype-specific information. Our analysis found that 85% of the genetic variants we extracted were shared across BD subphenotypes, indicating common genetic vulnerability. The plot shows the proportion of genetic variants from GWAS shared across subphenotypes versus those unique to individual presentations. The bar plot shows the number of unique variants. Their total identified variants are shown in the set size, and their shared variants are indicated by the dotted lines. We identified up to 181 subphenotype-specific variants, 50 of which were novel to BD. 9 of these were previously associated with related traits such as ADHD, dyslexia, substance use disorder, risk-taking, insomnia, neuroticism, cognitive function, inflammatory and autoimmune disorders, including thyroid disease. This further highlights the presence of both shared and unique genetic factors affecting the diverse clinical presentations of BD. We investigated the functional implications of genetic variants across subphenotypes through biological pathway enrichment analysis. The green heat map illustrates the differential enrichment in biological pathways across the subphenotype-specific GWAS. We also investigated subphenotype-specific enrichment in brain cell types, shown in the purple insert, and associations in specific brain regions in the grey insert. Our analysis highlighted key biological pathways, including synaptic function, neuroplasticity, and neural signaling, which were differentially enriched across BD subphenotypes and in line with the PCA clusters. Cluster 1 with BD1 was strongly enriched in postsynaptic organization and function. Cluster 2 with schizoaffective type and psychosis also showed enrichment in synaptic pathways, indicating shared biology with BD1, but enrichment in different molecular mechanisms. Cluster 3 with intermediate comorbidities, substance use and suicide, showed enrichment in synaptic plasticity and ion transport pathways. Cluster 4, the distant comorbidities of OCD, panic, rapid cycling, and unipolar mania, displayed more diverse enrichments, particularly in postsynaptic components and ion channel activity. Gene associations in brain tissues varied across subphenotypes, implicating varying regulatory mechanisms across brain regions and developmental periods. Rapid cycling contributed most and BD least to the variance in comorbidity in our variant PCA biplot. The Venn diagrams illustrate the difference in variant overlaps between these two subphenotypes and the other subphenotypes. Across our analysis, rapid cycling shared high risk for anxiety-related variants relative to BD1, yet the genetic correlation between BD1 and rapid cycling at the global level was strong, around 88%, which was similar to the strength of correlation between BD1 and BD2. However, locally they have distinct genetic architectures revealed by a multi-dimensional, more fine-grained approach. This supports their genetic separability, with rapid cycling potentially serving as a biomarker for anxiety-related conditions in BD. This last slide highlights ways in which both biclustering and GWAS enhance our understanding of BD by modeling it as a spectrum disorder rather than a single entity. Biclustering identifies novel subgroups based on shared genetic patterns, independent of existing clinical classifications, which may reveal unrecognized subphenotypes and improve risk prediction. Various algorithms can produce bespoke results. Irrespectively, biclustering and our current findings show a similar pattern when dissecting the BD phenotype. In future, integrating a genetically driven approach may enhance the specificity of individuals included in subsequent subphenotype-specific GWAS. Likewise, more robust clinical data could potentially be used to further constrain genetic data-driven algorithms. This current list of potential confounders in our BD research represents areas of ongoing research. Hopefully, these can be fully addressed in future analysis to increase stratification power further. In summary, our combined genetic and clinical analysis identified 4 distinct subphenotype clusters within BD. This enhances our understanding of the disorder's complexity and provides support for a partial underlying genetic basis for BD heterogeneity. Future research could focus on incorporating genetically driven data for GWAS selection and methods to address potentially confounding factors. Hopefully, this could lead to improved detection and early intervention in bipolar disorder. Thank you for your attention.
Great. Thank you so much. Can we hop right into the next speaker, perhaps? And we'll save the questions for the end.
There, okay. Yeah, so hi everyone. Thank you for being here, for your attention, and also I appreciate the opportunity to speak on some of my work today. My presentation is characterizing and utilizing phenotypic heterogeneity in bipolar disorder. I'm Katie Scott. I'm a PhD candidate at Dalhousie in Halifax, Canada, and I'm co-supervised by Dr. Martin Alda and Dr. Abraham Nunes, which we'll hear from later. So the 2 studies I'll be talking about today are the phenotypic clustering of bipolar disorder supports stratification by lithium responsiveness over diagnostic subtypes, which is an already published paper. And the second one I'm currently calling is exploring phenotypes of bipolar disorder in families with principal component analysis. And that manuscript is currently in preparation, but I'll be sharing some of the preliminary results today. So getting into the first study, it's open access. So if anyone wants to get more details about it later on, you can find it and have access to it. But essentially, the aim of this study was to examine 2 options of stratifying bipolar disorder and then see whether or not clinical data of bipolar disorder is actually informative of either variables. And these variables are the traditional diagnostic subtypes, so bipolar disorder type 1 or 2. And then also the alternative stratification variable is lithium treatment responsiveness. So we have lithium responders and non-responders. We've kind of already talked about the heterogeneity of the disorder and why we're trying to utilize that heterogeneity or kind of tease it apart in different ways. With the diagnostic subtypes of bipolar disorder, there's continued debate regarding the validity of these subtypes, and some have previously suggested that valid subtypes of bipolar should be based on clinically relevant outcomes such as treatment response. So that was part of the rationale behind us looking at lithium treatment response as a method of stratification because it has obvious implications for treatment response. So even within the diagnostic subtypes of bipolar 1 and 2, it can look very different from case to case. We have this highly variable presentation, different features, like some people might have psychotic features, other people don't. Maybe they have an earlier age of onset than usual. Some people might experience mostly depressive episodes, whereas others are primarily only experiencing hypomanic or manic episodes. So there are a lot of features that come together to make up the clinical picture of an individual who has bipolar disorder. So in this analysis, this paper, we used a clustering analysis on clinical data from adults with bipolar 1 or 2. And we also had information on their lithium response, which was came out of what we call the ALDA scale. And we wanted to see if clusters of this clinical information were informative on the labels of diagnostic subtype or lithium response. So we had 4 kind of general steps in the whole analysis, I guess, pipeline. We started with imputation of the data because we did have some missingness. And then we moved on to conducting a hierarchical clustering analysis. We then calculated adjusted mutual information scores to measure the information that was contained in these clusters about each variable of interest or the labels. And then finally, we did look at univariate comparisons of the phenotypic clusters, or perhaps clinical profiles. I'm not going to go into too much detail. People who are watching this later can kind of pause to see, but we did choose to use hierarchical clustering because it would fit best with our mixed type of data. And also we didn't have to specify the number of clusters we were looking for ahead of time. We maximized silhouette scores to figure out the optimal number of clusters. And then I had previously mentioned the adjusted mutual information score, which was a measure that we used, which essentially just quantifies the amount of information that one variable contains about another. So in our analysis, we measured the amount of information that the clusters contained about either bipolar diagnosis or lithium responsiveness. So our sample, we had 477 people. There were over 30 variables. We wanted to have as many variables as possible to try and characterize kind of the clinical picture of people. And you can see the totals there of bipolar 1 and 2, and then the lithium responders and non-responders. So we identified the optimal number of clusters was 2 clusters. And when we calculated the adjusted mutual information scores, we found over our imputed data sets that consistently the phenotypic clusters contained more information about lithium responsiveness than they did the diagnostic subtypes. And when we looked a little bit closer at these 2 clusters, we realized one cluster is quite a bit smaller than the other one. So this is the pooled results, and cluster 1 only had 56 people, whereas the second cluster had 421. And the first cluster was the one, it contained significantly more information about lithium responsiveness, which we can see on the next slide with right here. This is our categorical kind of label for the lithium response. We do have more of the continuous scores from the actual ALDA scale as well. But the general kind of main takeaway is that cluster 1 had proportionately way more responders. It was over 80% responders in that cluster, whereas only about 25% responders in cluster 2. And another slide just for people to see later on recording if they're interested. So the phenotypic clusters that were generated from clinical data, so data-driven clusters, contain more information about lithium response than diagnostic subtype, and the clusters showed the differential lithium response when we looked at the univariate comparisons. So it seems that lithium response and its associated features appear to account for more clinical variation than diagnostic subtype. And I chose to speak about this study first because the limitations of this study helped inform the approach that we used for the next one. And before I get into that, I also want to make it clear that I'm not saying to disregard the diagnostic subtypes completely. Like a subtype based on lithium treatment response may provide a better stratification of bipolar disorder in regards to research, because there's like evidence of the biological basis in prior literature, like cell studies, and then genetics related to response. However, the current diagnostic subtypes of bipolar disorder are still useful in clinical practice, since clinicians will often consider a diagnosis when choosing to prescribe, say, antidepressants as part of treatment, since it's reported that antidepressants are less likely to cause treatment-induced rapid cycling or mania in bipolar 2 versus BD1. And also the existence of bipolar 2 is kind of like a reminder for clinicians or health professionals to probe for any history of elevated mood episodes in individuals who may present with depressive episodes, because say in bipolar 2, like patients tend to be less likely to seek treatment for hypomania, so it can reduce the misdiagnosis of major depressive disorder in those cases. So onto the actual limitations here. One of the limitations we encountered was that we could only use Canadian data. And this was because when we tried to include some international sites, like we had European cohorts, they ended up clustering on their own. So they weren't like meshing with our Canadian data. And it wasn't very informative. And also the type of clustering hierarchical, we can't directly apply or translate it to an external sample. And we also didn't have a replication sample in this case. And then finally, we're still kind of having this issue of lumping the phenotype of bipolar disorder into just two groups or labels rather than characterizing phenotypes in a more continuous space. So we figured there were a lot of small effects that were contributing to the clustering here that weren't observable even in the univariate comparisons. So in the next study, we favored using principal component analysis for mixed data to tackle some of these limitations, and I'll talk about that more now. So this is the study I'm calling Exploring Phenotypes of Bipolar Disorder in Families with Principal Component Analysis, or PCA. We were focusing on examining the familial similarity of bipolar disorder phenotypes, because while we know there are genetic underpinnings of bipolar and they're quite strong, we don't actually know the parts of the clinical picture that are linked with genetics. So to explore this, we looked at the similarity of phenotypes in families. And for anyone not familiar with family studies, we use the term probands to refer to the first registered individual with the condition of interest, and they're the reference point for the relationship to other family members. So we went in with two hypotheses. We felt that probands and the relatives would have a more similar symptom structure or presentation compared to unrelated persons, and also that the close relatives of probands, like immediate family members, would be more similar than the distant relatives or controls. So our sample consists of adults, again, type 1, type 2 bipolar disorder. We had quite a few variables. There was less than 20% missing, and you can see the numbers there. And for the data used, so I have more of a description of the variables included, and then relatedness was measured or quantified using the coefficient of relationship, which essentially corresponds to the average proportion of shared genes. And there's some demographic information. So for a very quick rundown of the analysis pipeline, we imputed the data to take care of any missingness. We had much less missingness in this analysis than the previous one. We also have a smaller sample. Then we used the proband data to perform principal component analysis for mixed data types. So we were reducing the dimensionality of the data set by transforming it into the lower dimensional space while trying to retain as much information as possible. And it was also to help address any potential correlation between our many variables by creating the new set of features or principal components. So after we fit the model with the proband data and we get their kind of mappings on the principal components, then we took the data from relatives and controls and we projected that into the PCA space based on from the probands. And then again, we extracted the coordinates or scores of those individuals as well. And based on those, we compared the relatives and probands, or relatives and controls and their probands, using cosine similarity, which is a measure of similarity based on vector angle, where a +1 cosine similarity means two vectors are very similar. -1 would be the opposite. 0 would be no similarity at all. And then once we calculated that similarity, we performed a mixed effects linear regression to determine the association with relatedness, and we were able to use family membership as a random effect in that. And also, I looked at the standardized difference of individual PC coordinates, PC coordinates between the probands and their relatives and controls. So I analyzed the individual principal components as well for similarity, in addition to like this overall kind of batch of components. So the fit based on the probands We ended up keeping 3 principal components. We were retaining components with an eigenvalue greater than 1, and those explained about 66% of the variation in our data set. And these are pooled squared loadings. So when we look closer at the 3 retained components, we can see that there's fairly clear kind of, I guess, groupings or how the components load on the variables. And I've just highlighted the loadings here that were 0.1 or greater. So the first component, it's primarily loading on the frequency of episodes variables. The second one is mostly associated with those age of onset variables. And then the third seems to have to do with polarity. You can't see the directionality of the loadings here because they're squared loadings, and that has to do with the mixed data that we're working with here. It has to be squared loadings, but the polarities tended to go in the opposite direction. So with that third component, depression at onset and bipolar 2 were in one direction. The mania at onset and bipolar 1 were going in the other direction, and so on. It was things that we would expect to see with those relationships. So when we got to the mixed effects linear regression, this is looking at the cosine similarities, so the mappings of relatives and probands and the controls on over all of those 3 principal components. We did find that there was an effect of relatedness where closer relatives of probands were more similar than like more distant relatives or the controls. I also have Euclidean distance here because I know it's a popular kind of distance metric, so I just included it for people who are more used to seeing that. But the way that metric works is it's how far apart things are, so that's why we're seeing like the coefficient for the Euclidean distance is negative, which means the closely related have a shorter distance to each other. So they're in line with what our hypotheses for this were. And then the last part, I had carried out looking at the individual principal components and kind of relationship between probands, relatives, controls. And this used a standardized difference. And what we found in our sample, at least, is that the first principal component, the one that relates to episode frequency, there was still an effect of relatedness. But with the following 2 components, the age of onset and the polarity type 1, we didn't have a significant effect there. So to summarize, we were exploring the similarity of clinical phenotypes of the probands, their affected first and secondary relatives, and how they compare to controls. Using PCA allowed us to explore overall kind of clinical picture or symptom structure based on the data we collected, and to account for the correlation between variables while balancing the amount of information that we retained. And the results from the regression supported our hypotheses for like the overall symptom structure being related or being associated with relatedness. And then with the future directions, one of the limitations here is that we don't have a replication in a larger data set, although I do have a larger sample to do that on, and I have conducted that already. I'm not presenting the information here, but maybe a little bit of a spoiler. It seems like things are looking quite consistent between our sample and the other sample, so that's exciting. But after this, I also want to examine the association of the components with genetic risk of various disorders and traits, like those that we can determine through polygenic analyses. So I hope this presentation of these 2 examples is 2 examples kind of regarding how we can use the heterogeneity of bipolar disorder. It can demonstrate how embracing the heterogeneity rather than simplifying it into rigid categories can provide more insight into treatment response and perhaps genetic basis. And just a quick little thank you to everyone that was part of that with me, and I'll hand things off to Dr. Nunes.
Okay.
Thanks
Thanks,
again.
Mm-hmm.
Kate. It's not letting me share the screen. I think, Katie, you might have to stop sharing first so that I can.
My thing disappeared. 1 minute.
And I apologize, my camera seems to not be showing anything but a black screen on my side. So I don't know if you can all see me, but if you can't, it might be for the better. Who knows? Okay, let me just go to the slides here. Can you all hear me okay? Perfect.
Yep.
Yeah, my apologies. I don't know what's going on with the camera there. And my slides are showing up full screen.
Yeah.
So in the interest of time, I think I'm going to move through this a little bit quickly. So I'm a psychiatrist and a clinician scientist in Halifax. I do computational modeling, but I also run clinical trials. And I'm going to zoom out a little bit from the very excellent analyses that were presented earlier to think about how we can design studies in order to better capture data that we would require to understand the origins of heterogeneity in bipolar disorder or other diagnoses. And so essentially what we've been trying to do with pharmacogenomics is mapping from a genotypic space onto a phenotypic space. Here a space is just, you have a phenotype and as a point, and if the points are closer together, those phenotypes are more similar. Same for genotypes here. But this mapping really involves translation across multiple levels of analysis. This is just one organized by spatial scale, and then there's an environment outside of the individual that is going to interact. And so one of the reasons why developing this mapping is so difficult, we've heard about it several times thus far, is that the space of phenotypes, not to mention the space of these the space of, let's say, behavioral phenotypes, not to mention the space of intermediate phenotypes, is extraordinarily large. Even if we're just looking at DSM-5 presentations, there are about 37,000 ways that bipolar disorder type 2 alone can present. Now, you can argue that there's overestimates. You can also argue that these are underestimates too. Regardless, the size of this space is massive. Furthermore, there's a mapping that's going to exist between genes, molecules, synapses, and so forth, mechanistically in order to generate the behavioral phenotypes that we observe. And individuals with these different behavioral phenotypes will interact with their environment. And to make things even further complicated, the environment is going to interact with all these levels. And so the phenotypes at each of these levels in this mapping could evolve over time. And the complication of this heterogeneity, I think everyone on this call is already going to be familiar with that, is that first, and perhaps I should have organized these in reverse order, it limits the accuracy of clinical prediction models. For instance, if we're trying to predict the lithium response, which is what these two plots indicate, let's say we train on phenotypic data from one site, and then we attempt to predict lithium response in other sites. So each of these rows is a different site, and this is patient level accuracy, essentially just 1 minus the prediction error. You can see that the results are very inconsistent. This is a very common problem across prediction modeling studies. We also have poor diagnostic reliability. We understand this already, most of us. And I think that additionally, the diversity in the presentation can lead to interactions with the environment that then further generates more variability. For instance, if you have someone who has relatively, textbook, classical episodic course, but then might have some mixed features, perhaps this individual gets put on atypical antipsychotics. Those atypical antipsychotics cause insulin resistance, and then we know that this is associated with many other features that can be generated in that phenotype over time. And so the nature of the phenotype can evolve as we observe the individual. So why is it that among a group of people with bipolar disorder, you get such variability? Ecologists study this a lot in how biodiversity arises in nature. And there are several different mechanisms. I think that two of them are the most relevant to us. One involves something called allopatric diversity generation, where you start with some population, and effectively what you get is a categorical split in this population. So in nature, this might be, a mountain range arises or a river develops or something like this that separates two populations. And then over time, they diverge. For bipolar disorder, we might have something like a group of people who have exposure to antidepressants and then another group that does not. And if we just observe these individuals' clinical courses as time goes on, we may see that the clinical courses diverge substantially. Another form of diversity generator is something called a parapatric diversity generator, where you acknowledge that there is some variation within that original population. Perhaps it's not categorical in nature, and perhaps it's not relevant at the start, but with time, the extremes tend to reinforce themselves. So perhaps there's some degree of personality variation, whereby some individuals might be more prone towards things like substance use, general affective instability outside of mood episodes and so forth, until over time you observe these individuals interacting with their environment and their phenotype changes and diverges from one another. So these are two mechanisms. This is not, a hard and fast set of laws by which diversity is generated, but it just helps us to understand what are the things that we would need in order to understanding bipolar disorder, how this diversity arises. And so a couple of years ago, we came up with a set of, let's say, desiderata for studies that would be able to tell us about diversity generation in mood disorders. And we call this fields. So F is for family studies. And I think we've talked a lot about family studies here, and the audience here would know far more about genetic studies than I would, and so I'll move over that. The I would be interventional studies. We talked about allopatric diversity generation. You need some kind of categorical intervention there. And so if you have a cohort of individuals who you can follow, and then at different times you might take a subsample of these individuals and expose them to an interventional study, let's say an RCT of some sort, and then they move back into the cohort, you can follow them over time. This would be one way to sort of simulate that allopatric diversity generation mechanism. I think we need to do a lot with respect to characterizing the environments in which people live. And this is because, as we all know, the disease does not exist within a vacuum. It exists within an individual who interacts with an environment, and that environment can feed back. Of course, I think it goes without saying that we need longitudinal studies as well. And multimodal phenotyping. I was talking about earlier how the condition can be characterized across many levels of analysis. And so I think we need data across these levels of analysis in order to develop very rich pictures of the bipolar phenotype. And finally, statistical and computational advancements. We've seen several already today, and there are a few others that I'm going to talk about in just a moment here. One thing I want to draw attention to is that CanBind, which is the Canadian Biomarker Integration Network in Depression, is a consortium across Canada and also the United States. The US sites involve leadership from Canadians as well. So I think the name is still accurate. And this site uses a lot of standardized protocols. We have multiple studies that run together. And I think it's doing a very good job of capturing many of the desiderata that I mentioned earlier. And so Dr. Fry, who's the overall PI of CanBind, and we have this very large team, he has something called the Enable platform, which is something similar to what is currently called the Master Observational Trial. It's a large registry of individuals with mood disorders who have a very detailed clinical characterization, a lot of biomarker studies, and they're followed for a brief period of time to get certain biomarker recordings. But alongside this platform, we have multiple trials, observational and randomized as well, including 18-month long follow-up study that we're conducting now as well. And individuals can move within these programs so that we can kind of generate snapshots as time goes on. So I think this is one exciting project that's happening right now that can help us get towards heterogeneity to some degree in mood disorders. And then I also want to mention the HPO and global bipolar disorder cohorts led by Dr. McInnes and several others. And I think this is a very interesting statistical computational advancement to help us pool data across cohorts, something that's a really big problem. And hopefully by doing that, we'd be able to generate the kind of sample sizes that we need in order to better characterize phenotypic and genetic heterogeneity in this condition. So this is essentially all I wanted to present today, sort of going away from some of the analytical topics that we've talked about and thinking about how can we design clinical data collection processes in order to generate that rich phenotypic data that Adi was saying is quite difficult to generate. And with that, I'll stop my presentation.
All right, great. Well, thank you, everyone, for sticking it out, and thanks to our speakers. I think now we have time for a short question and answer session. There's a few questions. You can see them in the chat, targeted towards the different speakers. I can just quickly address one of the questions that, say, the first question that showed up at the beginning. And one way to interpret that question is, when I take this big high-powered PRS score and test it on this BDRN subgroup, how do I know that I'm not just getting lucky? And by looking at the bicluster patients in BDRN, how do I know that I'm not just maybe by chance obtaining a better PRS score than the global 72%? We tested for that specific question, assuming that's the question that was asked. We tested for that specifically by running a bunch of permutation tests shown in black here, where we randomly subselected different random collections of case subjects with the same size as each iteration would otherwise indicate. And we can see that the AUC in the real data, which is higher than the global 72%, is quite a bit statistically significantly higher than all the random shuffles. So we would give this a p-value of 0.00, forget, 0.001, 0.0003, something like that. But anyway, that was just a, let me stop sharing my screen and bring up one of the other questions here, which is, I think the second question was directed to Tracey. And if you can't see it in the chat, I'll read it out loud. What were the clusters identified in the plot with the psychosis and comorbidity axes? Were those resulting from previous factor analysis, or did you perform separate clustering analyses? And if the latter, what methods did you use to obtain the clusters? Some of them overlap a lot.
Thanks, Adi. It wasn't designed to overlap. The first set of clusters is just clinical data from factor analysis. And then the second plot of the PCA, that was using the variance from GWAS of the subphenotypes, the individual subphenotypes.
Okay, excellent. Well, thank you. And then to Katie, a question. Yeah, there are two questions. The first is so principal component analysis is often used as a proxy for developing covariates that are associated with ancestry. Do you think that the principal component analysis that you're applying to the phenotypes captures, is maybe driven by the same ancestry, or is it capturing other effects?
Yeah, so I can actually, I guess, maybe not answer that question to as much as you may hope, but the idea for using principal component analysis in this case was more to be able to convert our mixed data into a more continuous landscape, and then also to reduce the dimensionality. So I'm not sure, I don't think it's necessarily measuring the same thing as when we use it for genetic analysis, but the idea of kind of comparing between relatives afterwards would be to examine the familiality if there is more of a genetic or ancestral kind of connection below. And I can actually super quickly answer my second question I see there, which is how much overlap there is between the lithium response groups and the bipolar subgroups or subtypes. And if you look at, you can see the full paper online, but in the cluster univariate comparisons, there was no difference between clusters in the distribution of bipolar 1 and 2. And also when we compared the sample before all the clustering, just stratified by bipolar disorder subtype or stratified by lithium response, we had the same number of responders and non-responders in both subgroups.
Wonderful. Thank you. Thank you again. I think we've hit the hour. So thank you for everyone for joining, and yeah, we can follow up by email. Thanks again.